Hello, my name is John Sparren. I'm pleased to chair this session, which is devoted to gene therapy and drug development. I serve as chair of the Scientific Advisory Board for the CMTA, and I'm really pleased to have really an all-star team for this session. We're going to have Dr. Cleopa, who's a neurologist and a leader in gene therapy efforts for CMT. And we'll have his presentation first. Then I'll just briefly introduce Mark Scheidler, who is chair of the Therapy Experts Board for the CMT Association and has developed a lot of company partnerships that he'll tell you more about as well. And also we'll have Maurizio D'Antonio, who's at Milan, and he's really pioneering a number of different new pathways for drug targets for CMT as well. So I'm excited to introduce these really great individuals to you, and I'm going to hand it over, I think, first of all, to Dr. Cleopa.
Good afternoon. Thank you for joining us in this session about our gene therapy program for CMT1X. My name is Cleopas Cleopa. I'm a neurologist at the Cyprus Institute of Neurology Genetics in Cyprus, and I will give you a short update on our progress regarding the development of a gene therapy for CMT1X. You have already heard about the disease, but just a brief introduction. CMT1X makes about 10% of all CMT patients, so it's actually the second most common type of CMT, and this is an X-linked disease which affects males selectively, more severely at an earlier age, usually starting in the first or second decade of life, and shows a slow progression over the lifespan. The symptoms are well known and present with difficulty running, ankle sprains, foot drop, sensory loss in the legs. Female carriers are usually asymptomatic or with a later onset after the second decade of life and with an overall milder phenotype. And there are some particular features for this CMT neuropathy. Although classified as demyelinating CMT, it shows intermediate slowing of the nerve conduction velocities and a mixture of axonal and demyelinating pathology in nerve biopsies. The genetic cause of CMT1X is the mutations in the gap junction beta 1 gene, the GJB1 gene that encodes the protein connexin 32. Connexin 32, as shown in this diagram, is a gap junction protein that forms channels between the Schwanzel that forms the myelin sheath and the axon that transmits the signals along the nerves. These channels are very important for maintaining the homeostasis, the well-being of the myelinating cell and the myelin sheath and the axon itself. More than 400 different mutations have been reported in patients suffering from CMT1X, and a lot of work has been done to understand how they cause disease, and all this work led to the conclusion that they cause the disease through a loss of the protein function. So our strategy for gene therapy to treat CMT1X consists of replacing connexin 32, so introducing the healthy connexin 32 gene. And because connexin 32 is expressed specifically in myelinating Schwann cells in the peripheral nerves, we designed the strategy to target specifically the replacement of the gene in these cells. And so in order to achieve that, we use viral vectors, currently adeno-associated viral vectors that are widely used in clinical applications for gene therapy and have an established safety profile. And we have cloned the connexin 32 gene under an element, a DNA element that drives the expression specifically in Schwann cells. This is a myelin-specific promoter. We also did a lot of work to optimize the delivery method for the viral vectors to deliver the connexin 32 gene. And after testing several approaches, including direct injection into the nerve, injection into the muscle, or intravenous injection, we now focus on lumbar intrathecal injection of the viral vector. This is introduction directly into the spinal fluid area in the lower back, which is a method that can be applied in the clinic, is already used in clinical trials, and provides adequate distribution of the viral vector along the spinal roots and along the length of the nerves, and therefore is suitable for the gene therapy application. So this is the design of the DNA construct that we use to deliver genes specifically to Schwann cells. This is the example of a vector that does not include the therapeutic gene, but includes a reporter green fluorescent protein gene under the control of the cell-specific promoter myelin protein 0, which restricts the activation of this DNA plasmid only in Schwann cells. When we package this DNA into the AAV vector and deliver it to experimental mice by intrathecal injection, we can demonstrate that we obtain widespread expression in myelinating Schwann cells. You see this red signal here, and in this picture, a green signal in many Schwann cells along the anterior lumbar root, but also more distally in the distal nerve of the limb, the sciatic nerve, both in the section and in individual fibers, in a high percentage of Schwann cells. This is the design of the therapeutic vector that delivers the human GJB1 connexin 32 gene under the control of the same cell-specific promoter. So this is what we use currently for the treatment studies in the disease models. Again, this is delivered by lumbar intrathecal injection, and we delivered it in mice that are deficient for their own connexin 32. They are knockouts. So in this background, we can see the expression of the virally delivered healthy gene. And you see here the example of a knockout animal that has not been treated, has no expression of connexin 32, as opposed to this animal here that received the gene therapy vector and shows expression of connexin 32 in paranodal areas where it should normally form the gap junction channels, similar to a healthy normal mouse. So based on this overall strategy, we then continue to perform a treatment trial in the knockout model of CMT1X. This is the work that has been funded by CMT Association and Muscular Dystrophy Association over the past several years. So we treated these mice both at an early time point in their life as well as at a later time point, because we want to demonstrate that we can achieve a therapeutic benefit both before as well as well after the onset, the beginning of the nerve pathology, which starts around the age of 3 months in this model. This is important because when we hopefully get to treat patients, we need to make sure that also patients at a later stage of their disease in adulthood will still have a benefit from this treatment. So we treated the mice either with a therapeutic vector or with a mock non-therapeutic vector and compared the outcomes using various outcome measures, including measuring the muscle strength, measuring the nerve conduction velocity, and also looking at the pathology of the peripheral nerve. Here you see the results of muscle strength testing with the foot grip test, and you can see both in the early treatment groups and in the late treatment groups, there is significant improvement in muscle strength in treated as opposed to the mock-treated animals. We also see significant improvement in the sciatic nerve conduction velocities. These are untreated mice. These are the treated mice early on. They approach the level of the healthy mice in terms of the velocities. And when we treat mice at the later stage, they improve significantly, but they do not reach the level of the normal mice, indicating that we should treat as early as possible. We also examined blood biomarkers that could be useful also in future clinical trials. The first one is the neurofilament light molecule that is detectable in blood at increased levels in patients with peripheral neuropathy. We showed that it is also elevated in the mouse model, and it shows a clear response to treatment. So it's a treatment responsive biomarker. Likewise, we identified another biomarker, the neural cell adhesion molecule 1, which also shows elevation in the disease model and clear improvement after treatment. So these treatment responsive blood biomarkers will be very useful tools also in future clinical trials to show a benefit of any treatment for CMT1X. Of course, we looked at the pathology and showed that there is significant improvement in the degree of myelin abnormalities in a distal nerve. The example here is a femoral nerve from a treated mouse, and this is the mock-treated, the control mouse. You see many more fibers that are demyelinated or have very thin myelin sheets, and this pathology is improved after treatment. Finally, we wanted to answer a very important question, which is whether our gene replacement therapy could be beneficial for patients with all kinds of connexin 32 mutations. And about half of CMT1X patients have mutations that cause retention of connexin 32 in the Golgi within the cell. We call them Golgi retained mutations. So we treated two transgenic mouse models expressing two of these mutations, the R75W and the N175D mutation. We treated them in a similar way as we treated the knockout mice, both before and after the onset. And we have completed the analysis of the early treatment. The late treatment study is still ongoing, but should be completed soon. What we have seen is that these mice that express CMT1X mutations show a similar response to gene therapy treatment. This is the nerve conduction velocities and the muscle strength testing of the R75W knockout mouse. You see significant improvement after treatment, and we see similar treatment benefit in the N175D knockout mouse. We also see significant improvement in the femoral nerve pathology. This is a treated R75W mutant mouse, and this is a mock-treated. Significant reduction in the number of abnormal fibers and in the number of inflammatory macrophages. So what I have shown you is that we have a proof of principle that gene replacement therapy provides a therapeutic benefit both before and after the onset of the neuropathy in 3 different models of CMT1X. We have completed the results of early treatment in the transgenic mice, and we are now completing the late treatment study. And also what's important, we have since last year an agreement with a gene therapy company to support the CMT1X gene therapy program. We use a different type of AAV vector, the AAVrh10, to deliver the human connexin 32 gene under the control of a human promoter element. So this is what we're currently testing in a project supported by this company, and we perform a dose escalation study. So we use different doses of the vector to find the optimal dose to treat the disease. And we also perform toxicity studies to ensure that there is no adverse effect of our gene therapy, something that we have not seen so far in our experiments. And we hope that by next year, this program will be further supported by this company so they can support further steps towards an IND application, including biodistribution and toxicity studies in larger animals. And this work will complete the preclinical package in order to be able to proceed, hopefully in the not so distant future, with a clinical trial. I would like to thank you for your attention and for joining us. I would like to thank the CMT Association and the Muscular Dystrophy Association for ongoing support of the CMT1X program all these years, as well as my team and my collaborators for supporting this work. Thank you. Thank you very much.
So I just want to mention that because Dr. Cleopa is in Cyprus, his presentation was recorded because it's quite late at night there. And but if you submit questions, we'll try to get those answered. However, I'm really pleased to welcome Mark Scheidler, who's worked with the CMTA for several years to spearhead development of company alliances. And he'll tell you more about those efforts across the board of CMT. He's also chair of the Therapy Experts Board for the CMTA. So please go ahead.
Thank you, John, for that introduction. What I wanted to talk about today was really the reasons why the CMTA has an alliance strategy, and also about the importance of working with different pharma and biotech companies to bring therapies forward. Excuse the technical issue here. So what is the problem we're trying to solve with having pharma and biotech alliances? Well, there are fundamentally, the real issue is that there are few new therapies entering testing in patients. And support the companies in terms of the expert knowledge and access to specialized CMT resources has been low for a set of related diseases with this many patients and no available therapies. So how do we go about changing this? Well, you need to understand that most drugs actually fail in clinical testing. So we need to build a portfolio of possible therapies to test. And in doing this, there are really three key areas that enable the efforts of drug developers. One is animal testing, and I'm going to show you some case studies that we've been engaging in. The second is clinical trial readiness, and I know John's commented this on in his earlier talk. And I'll finish with how we're going about involving patients in the discussion. Now, in order to have the biggest impact and treat the most patients, the CMTA is going to need to attract the interest of companies with the resources and expertise to advance a new therapy to patients. Once you've gotten something into clinical testing, the amount of resources required to actually get a drug approval and all the expertise involved in doing that is really their domain. And so for that reason, we really need to work towards getting into discussions with companies that would enable the development of a CMT project in their domain, applying all their expertise to solving the problem. I'm going to focus really on solving the first problem, which is animal testing. So when a company's developing a therapy at the early stages, there are usually a choice of diseases where they think that therapy may be useful. So our job is really to convince them to give a go in treatment of a CMT disorder. And we can do this by pointing out the frequency of CMT in the population is relatively high. It's really one of the least rare of the rare diseases collectively. And secondly, that there's a really firm knowledge of the disease causation in these different CMT disorders. And that discriminates these disorders versus a lot of other disorders that pharmaceutical companies could be working in. They then need to be able to show that the therapy works in an animal model before proceeding to testing in patients. And that's really where we can help them because there's a lot of knowledge and a lot of time and effort invested in setting up program to do animal testing in validated CMT models. So to provide them with this ability, the CMTA has invested in a preclinical testing platform that allows companies to access well-characterized models of CMT for drug evaluation by a network of trusted experts. And this involves a lot of the STAR Alliance consortium in terms of the knowledge of the sponsored investigators and the advisory board members the CMTA works with, as well as a network of service organizations that are really expert in doing the sort of evaluation and testing that we'll need them to do. So this gives you a little snapshot about how we got into this, what our initial relationships were. And I'm not going to drill down into the data or the results of this, just really a snapshot to show you that we started off this strategy back in 2014 through the formation of a relationship with Sanofi and Genzyme, and also another relationship with Ionis. Since then, we've really developed a really strong portfolio of alliance partners year in and year out. Several of them are shown on the bottom of the screen. And I mention those because they have an interest in CMT type 1s. We also have a number of alliances in type 2 CMTs. And secondly, because these companies have engaged with us in terms of releasing information to the public about the collaboration. And they've also participated in video interviews that we've done with them that are available through the CMTA that we published last month. And the CMTA can refer you to those interviews, which go through a list of questions about what patients may be interested in knowing about what the specific company is doing in the development of therapies. So let me go through quickly a couple case studies of work that we've been doing with different companies, kind of what the target is, how we're going about doing it. So the first case study I'm showing here is with Ionis. And this was a joint effort to design and test antisense oligonucleotides. I know it's a mouthful, so we just call them ASOs, in CMT1A mice and in CMT1A rats. So you need to think of ASOs as a piece of sticky tape that blocks a genetic sequence that's responsible for producing the PMP22 in Schwann cells. And, people with 1A are well aware that making too much PMP22 is tightly associated with the development of the disease. The results from this study, which have been published, showed that this approach can block progression of CMT1A symptoms. And they also suggested that this therapeutic approach could possibly reverse the course of disease. Let me show you a second case study, which is a fairly new one with a Swiss company called Adex Therapeutics. Now, their approach is to treat CMT1A by targeting Schwann cells with what is called a GABA PAM. Again, I know it's a mouthful. So in simple terms, this is a drug that targets a receptor on the cell surface of Schwann cells that has been shown to modulate the expression of PMP22 in those cells. So think of this is a lock and a key, that there's a door on the surface of the cell and a lock on it, and that lock is a receptor, and there's a normal key that fits into it to open the lock. And that natural key is a chemical called GABA, which serves to control signaling between cells in the nervous system. And the Adex approach is to try to make a small molecule drug that controls how successful GABA is at opening that lock. So the joint studies with the CMTA and Adex are using CMT1A rats to confirm that their drug candidate can control the amount of PMP22 in Schwann cells, and that doing so reduces the development of the disease. An entirely different approach is being taken by a company in Korea called ToolGen. So ToolGen since 1999 has been a leader in Asia in designing and making tools that edit the genetic code. One of their founders is one of the original inventors of an early approach to do this. So the current promise of gene editing approaches like CRISPR-Cas9 as gene editing approach led them to go beyond providing biological tools to actually developing therapies. And they reached out to the CMTA to help them form a testing alliance to be able to test drive an approach by which they wanted to see if they could do this. So this effort is aimed at editing the sequence of the gene responsible for producing PMP22, such that cells are corrected and produce normal amounts of this protein. We're using both CMT1A rats and mice, and we're infecting them with a virus containing the editing tools. Animals are then assessed to see if they can produce normal amounts of the protein, and if they do they also correct the disease effects that we see in those animals in terms of changes in balance gait and grip, changes in the electrical signaling in the nerve, changes in the histology, that is the apparent, the normal structure of a nerve that we can see in a microscope. So I want to finish by mentioning that where you want to go with this, that we really want to facilitate companies to get enough proof of principle information that would accelerate movement of a therapy candidate into clinical testing in humans. And Farnex is a French company that has done this with what I would call a first generation of the GABA agents that I talked about just now with Adex. And they're developing a first gen treatment for 1A, which involves combining a known GABA modulating drug with other agents that improve its function. It's an old drug that's well known and approved for another use. And by combining with other agents, they're able to turn its function towards Schwann cell receptors that are controlled by GABA. This drug combination has shown positive effects in clinical trials in slowing the progression of 1A symptoms in patients. And we're actually, I think the community is very excited about the potential of a first generation therapy being with patient access. In order to help them to get across the goal line, Farnex has engaged CMTA's patients as partners advocacy efforts in order to help them to refine the presentation of the data in terms of what it would be useful for, what the interest in the patients is in terms of solving their problems, and take that input and use that to help them to bring the drug to market. And with that, I think I'll close there and turn over to the next speaker, John.
Thank you very much. So for those of you who may have questions, I think we'll hold them until after the next talk. You can also put them in the chat if you want to. So for our last speaker of this session, I'm really pleased to welcome Maurizio D'Antonio. As I mentioned, he's really an expert in Schwann cell biology and has worked in CMT1B and also CMT1A for several years. Let me just let him go ahead and give us his update. Thank you, Maurizio.
Okay. Thank you very much, John, for the introduction. I just want to make sure you can actually see my screen.
Yep, it looks perfect.
Okay, that's great. So, hello, everybody, and good afternoon or good evening for those like me are actually in Europe. What I will be doing today, I will be actually give you an update on CMT1B, and I'll just try briefly to give an overview of what CMT1B exactly is, what is the current knowledge on the molecular mechanism of the disease, and the ongoing research that we're actually carrying on with a lot of support from the CMTA for the identification of potential therapies. Now, you may have already heard in a previous session from Mike Shai that CMT1B is caused by mutations in the myelin protein 0 gene. And you can see here in the left side the structure of P0, that is a single pass transmembrane protein, which has a large extracellular domain, which is particularly important because, as you can see here in the middle picture, it is necessary to form these tetramers of P0 that allow the compaction of the myelin sheath. In the end, what you have is, you can see here in the top image, a nice axon in white, which is surrounded by the myelin sheath in black, which is really nicely compacted. What happens is that when the myelin protein 0 is not present, and this can actually be achieved in mice by literally ablating the gene in the mouse. When P0 is not present, myelin loses completely its compaction. And this told us back in the 90s that the main function of P0 is actually to compact myelin, insulate and protect the axon, and allow fast conduction of nerve impulses. Now, however, as has already been mentioned again by a previous session, the problem we're facing with mutation in myelin protein 0 is that we can actually have two forms of the disease, which are quite different. We have a first form, which has a early onset, which is either infantile or really in childhood of CMT1B, where you have delayed motor milestones, severely reduced nerve conduction velocities, and in terms of morphology of the nerve, you have either dis or demyelination, meaning that either myelin is not properly formed or it's formed and then destroyed. But in reality, and rather surprising, there are also a series of mutations that gives rise to a late onset form of CMT1B, which is also known as CMT2J or I, because this mutation in reality cause a damage which is mostly in axons rather than in myelin. And indeed, the patients have the disease late in life, so they normally reach a motor milestone normally. They have normal nerve conduction velocity, but then they start losing axons, and that's when the problem arises. And the two pathologies are so different that we do really believe that the mechanisms are different between these two subtypes. So I will start just giving a brief overview of the first project, which has been also partially funded by the CMTA, which is the identification of modulation of a process which is called the UPR, which stands for unfolded protein response, for the treatment of early onset forms of the disease. To do this, we have generated in the past mice carrying mutation, which are also found in humans. In particular, this mouse you can see here is called the myelin protein 0 serine 63 deleted mouse, meaning that in the P0 of these mice, we have lost, we have lost the serine, the amino acid serine 63 in the extracellular domain. As a result, the myelin in this mice is thinner, and you also have myelin loss and formation of what are called onion bulbs here, shown in the arrow. But what happens is with protein is that the protein doesn't reach the myelin sheath, but is completely retained in the endoplasmic reticulum of the cell. So basically, instead of reaching the membrane of the cell, is retained intracellularly. What happens when this happens is that the process of folded protein response is elicited, and this is due to the fact that there is indeed protein in the plasma reticulum, which causes stress. These three pathways, which are called IR1, ATF6, and PERK, are activated. The function of PERK in particular is that of phosphorylating this factor called CF2 alpha, which reduces the amount of proteins which are made, of course, to try to solve the problem of having protein retained in the ER. The other two pathways, IR1 and ATF6, generate transcription factors, which are here XBP1S for splicing, or ATF6N, nuclear ATF6, that then drive the expression of genes which are involved either in protein folding or in protein degradation. Basically, because what you want to do is either to manage to fold this protein which is retained in the ER and send it to membrane, or to just eliminate it. What we have shown in the past is that the treatment with a molecule that modulates the PERK and the phospho-CF2 alpha pathway of the UPR is able to largely ameliorate the pathology in the serine 63 del mice. This work has already been published, so I will not spend too much time on it, but it is important to underline that more recently, and in collaboration with Mike Shai and Inflectis Bioscience, which is the company that developed CEFIN1, we have also treated our mouse models of CNT1B, specifically the R98C mouse, and mice that recapitulate CNT1A, and we also had benefits in these mice with the treatment with CEFIN1, and this work will hopefully be published soon. And right now, CEFIN1 has already completed phase 1 clinical trial in humans, and a phase 2 clinical trial is being planned, hopefully for end of 2021 or early 2022. But at that point, we also started wondering, okay, we may have a molecule that could work, but as Mark told us before, of course, we cannot just rely on one single molecule. We also need to have alternative strategies and approaches to make sure that if one molecule in the end does not work, we have alternatives. And to do this, what we set out to do was to figure out what the role of the other two pathways of the UPR were in CNT1B. And as you can see in these images here, very briefly, you can see on the left panel that when we remove the factor XBP1 from serine 63 del mice, so from CNT1B mice, the phenotype of the mice dramatically worsens. And if you now look on the right side, again, this is very similar to what happens when we eliminate ATF6 from the CMT1B mice. The phenotype largely worsens, and this can also be seen by measuring nerve conduction velocity. As this was telling us that these two factors, so XBP1 and ATF6, are so-called adaptive in the neuropathy, meaning that we need them to be there, we need them to be activated during the unfolded protein response to try to protect the Schwann cell from demyelination. So what we set out to do now with the support of the CMTA was also to look whether it was possible to revert the CNT1B phenotype of these mice by increasing the expression of these genes. And we have done first this by using mice that are able to overexpress one of these factors, namely the XBP1 factor. And as you can see here, myelin in the mice, CNT1B mice that overexpress XBP1 is much thicker, the mice are performing better, and also nerve conduction velocity is improved in these mice, which clearly shows that it is important to increase the expression of XBP1, the signaling through XBP1 to improve the phenotype. And what we are doing now, in collaboration with the Scripps Institute in California, we have identified small molecules that are able to specifically activate XBP1. And right now, experiment in vitro and ex vivo have shown amelioration in myelination, and similar results has also been seen when we use small molecules that specifically activate ATF6. And therefore, right now, we're planning to test these molecules in vivo in CMT1B models, but potentially also in other forms of CMT, such as CMT1E or CMT1A as well, where there are alterations in what is called cellular proteostasis, and therefore there is the activation of pathways such as the unfolded protein response that I've shown you a few minutes ago. Instead, very briefly now, what about the late onset kind of CMT1B? Well, this is, as I said, a very unique case where you have mutation, which is in myelin, but the problem appears to be in the axon. If you take biopsies from patients with this disease, myelin appears to be pretty normal, as you can see in the panel in the top right, but there are very few fibers, meaning that their axons are being lost. To study how this is happening, we have generated, in collaboration with Larry Rabbets and Laura Feltri in Buffalo and Davide Parison in Milan, 2 mouse models for late onset CMT1B. The mouse carrying the threonine 124M mutation or the P70S mutation. And what we have seen is that these mice do replicate some of the features that are seen in humans. This is important because it's actually the very first mouse model for this late onset forms of CMT1B. We see the neurophysiology is impaired, and you see here in the morphology in the right panels that there is loss of fibers and the degeneration of myelin. And if you see instead in the lower left panel, where you can see these green images, you see these blobs, which suggest that in these mice, axons are being lost. And so this means that we are actually recapitulating the disease. What is important is that it appears that one important factor that has been identified in many neurodegenerative conditions, which is called SARM1, appears to be activated also in these mice. And we can actually see that because some metabolites in these mice are altered as compared to normal or so-called wild-type mice. For example, just a couple of them, this factor, which is called NAAD+, is reduced, and ATP production is also reduced in these mice, which are usually considered to be hallmarks of activation of the SARM1 pro-degenerative pathway in the mice. So what we are doing and what we have done in reality, we have looked at what happens if we remove SARM1 from the threonine 124 mice, and the results are promising because we have started seeing a reduction of the number of axons which are actually degenerating in these mice, suggesting that at least partially elimination of SARM1, the genetic elimination of SARM1, is able to protect these mice from degeneration. And at the same time, I'm sorry that here, unfortunately, this is not showing you really properly, but you have also heard a lot how it is important to identify biomarkers of disease. And we have shown that in the CMT1B mice, especially in the late onset forms, where we have axon degeneration, we have an increase of neurofilament in the plasma of these mice. And after treatment, not treatment, but crossing these mice with the SARM1 null mice, although it's not shown here, so I hope you will believe me, there is a reduction of plasmatic neurofilament, suggesting that the pathology could actually be indeed ameliorated, but also importantly, that neurofilament could represent a treatment-responsive biomarker to be used in clinical trials in patients. So just to conclude, I hope I have shown you how we really have to think about CMT1B as two distinct diseases, basically. One early form of CMT1B with primary either dysmyelination or demyelination, severely reduced nerve conduction velocity and delayed motor milestone, and another form which is late onset, so usually between the third and the fifth decade of life, with primarily axonal degeneration. The mechanism of disease appear to be really different, and we have identified, at least in part of the early onset CMT1B, protein misfolding accumulation in the endoplasmic reticulum and activation of the UPR as a very common event, whereas in the late onset forms, although the mechanism is still not clear, the mechanism leading to axonal degeneration, we think that it really has a lot to do with axon-Schwann cell communication. For the early onset, we have gathered data suggesting that compounds that do modulate the unfolded protein response are appealing candidates for treatment, whereas molecules that might actually limit axonal degeneration, such as SARM1 inhibitors or maybe even HDAC6 inhibitors, may provide benefit for the late onset forms. And with this, I thank you for your attention, and of course, I will be happy to take any question.
Thank you so much, Maurizio. We do have a little time maybe for a question or two. We have about 5 minutes left. I can look through the question period, but also I think if you have the ability to unmute yourself as well, you could just ask it as well. And feel free to ask a question for Dr. Scheidler, and also if you have a question for Dr. Cleopa, we might be able to answer for him, although his presentation was a recording. One of the questions that was in chat is the 1X therapy that Dr. Cleopa had talked about. Is this also applied to other types of CMT? And he's certainly working on other types of CMT. The details of the gene therapy will be a little bit different, but he's been working on CMT1A and some other types of CMT as well. So he's been working on with really several different models of CMT. So there was a question about the Farnex trial, about it, whether it had formulation issues, and there was a response from Mark. Do you want to say a little bit more about that, Mark? Or?
Yeah, so the formulation issue, as it's called, was what's called a CMC issue. It was essentially a manufacturing problem with the higher dose. And Farnex does want to take the higher dose of 2 doses they tested into registration for patient use. So for that reason, the FDA required them to repeat their work with a manufactured lot of the drug that they had approved. So they're just doing this with the remanufactured higher dose formulation of their Pleo drug, as it's called.
So, Mark, another question that appeared in the chat was, has the CMTA attempted to form open science partnerships? These typically accelerate drug discovery and development by foregoing profits and patent protection in favor of public data sharing.
Those type of relationships tend to be very effective at a early translational stage, where there's general knowledge to be benefited from being shared with the community. John, I think that your biomarkers effort fits that kind of open science approach. When it comes to actually therapy development, if something's not patented, it's not going to get commercialized. So companies tend to take the view that if we have a novel composition, whether it's a gene therapy or a biological or a small molecule, if you haven't protected its commercial ownership, then it's very hard to convince anybody to put any money into it to get it through the very costly and long development and out to patients. So I've kind of broken it into two areas. There are a number of areas where open science can get done that benefits everybody, and kind of outcome measures clinically and biomarkers are definitely one of those areas. Another new area that's kind of coming up is what I would call target identification and validation. We need more targets to make drugs against for CMT, and this requires that we identify what the best targets for drug development are, and then show rigorously that they are good candidates for developing a therapy. And I think that's a really big area emerging in open science.
So I would just mention that most all CMTA-funded projects are urged to publish their results as soon as they're ready for publication. But with company efforts, certainly they have the decision on their own. So I believe,
And we have, I'd say, John, we have done both within a collaboration. So with Ionis, they were very interested in the ability to commercialize a therapy. But at the same time, they were very open to publishing the marker evidence, and also the tool compounds that we used in that study that will not be clinical development candidates were actually published together with the CMTA.
So with that, I believe we've reached the end of our allotted time. I want to thank the speakers again, Dr. Cleopa and Mark Scheidler and Maurizio D'Antonio. And I appreciate everyone's involvement in this session. I think the next session will be starting soon, so I encourage you to join the appropriate session next time. So thanks again to the speakers, and I really appreciate all the participation today.
Well, thanks a lot for everybody joining.
Thank you. Bye-bye.